But I already texted him and said, thanks for
I feel good about the presentation part. The after part, I think we're stressful, for sure. And I've presented most of the stuff multiple times. How you doing? Get him, man. Thanks.
Well, you know, partners paid, you guys did.
We'll be fine.
Wait and see.
Perfect.
Yeah, sir. That's creamer? Oh, yeah. Anybody need creamer? Yes.
Did you pass?
Pass.
Okay, I think we're just about ready to get started here. Find a seat. This is a new one for me. I have yet to have been in a defense in the hybrid building. So Bryan threw something new at us. So Bryan, it's my honor to introduce Bryan today. He was co-advised by myself and Jeremy, but I'm assuming that I'm introducing him because Jeremy's sitting down there. Bryan was born in Maryland.
Oh.
Oops, wrong one, sorry. Yeah, I had to look it up. He's a very cute kid, as you can see here. He's having sort of maybe a spiritual moment, telling his teddy bear a little secret. He's a messy eater. Hopefully, you probably grew out of this. He's the son of a diplomat. If you don't know that, I'm sure that they trained him out of this sort of messy behavior. But this is the first picture that I found really sort of disturbing. So he is the son of a diplomat. And here he is. He's got so many weapons. So he's naked, almost. And then here he has a sword, and then he's also holding an automatic rifle. Here he is actually in Jordan with his parents, and he has two swords and then assorted weapons that you can't really tell what they are in this picture over here. So a little creepy. I always thought of him as kind of a peaceful person. He stayed in Maryland and he did his bachelor's at the University of Maryland in environmental science, policy, and conservation, a really, really long name. And while he was there, he had a lot of really great and very, very diverse undergraduate research experiences. He went over to the University of Idaho at Moscow, where he studied macrophytes, milfoil, and DNA. That was like his sophomore year, maybe. Went all the way around the world to Japan, where he studied and mapped trees in traditional shrimp ponds with the World Wildlife Foundation. And then after he graduated, he headed out to California, where he was the crew leader for a trail maintenance and ecosystem rehabilitation crew. And then he did one more little jump up to the Tetons, where he did some more trail maintenance work before he came here and he interviewed at USU. He's an avid outdoorsman, and so here you can see himself trying to kill himself in Moab, and then again trying to kill himself in Logan Canyon. And when he interviewed, Colton is my most, where's Colton? It's always my most sort of severe critic of the people that we interview when they come. And Bryan must have asked too many questions about the outdoors because Colton concluded we definitely don't want to give him the position. He just thinks that graduate school is about rock climbing and mountain biking. But I would like to note that Haley, and there's a little foreshadowing there, wrote back and said he did have the shiny shoes on, and that meant he meant business. He's got so he means business. I think he needs to stick to more summer sports. I learned that he really never took to skiing or snowshoeing. But all that joking aside, he was incredibly committed to his project. He actually, in fact, managed to combine mountain biking with sampling. So here he is with his waders, his mountain bike, and his stadia rod. He was a very dapper little dresser as a youngster, but actually that's that diplomat influence again. But then things just sort of really fell apart. Any of you who know Bryan know that he can show up in some really, really strange things. I don't know if you can see this whole disturbing, I'm going to use the word package, but this actually says icebreaker, and I don't know what's going on there. It's just too much for me. And then this is a shirt that is covered with about 500 burrs, sort of old-fashioned statement, which he apparently had to burn afterwards. He's always been very dedicated to safety. So here he is washing the dishes with some goggles on in case he breaks a glass. But a very reoccurring theme in the College of Natural Resources seems to be peeing outdoors. Of course, we probably all do that a lot. But more importantly, liking having your picture taken while you're peeing outdoors. Here he is sampling out the toilet. This toilet got delivered to Stephen Klobuchar's office because he's such a pig, they decided he might as well just have a crapper in there as well. But in here's probably one of the best ones. Here Bryan is dressed for Halloween as Care Bear. So this is a Care Bear outfit that he's managed to squeeze into because some of us affectionately call Gary Gear Bear, to be dressed as Gary for Halloween. And he's here with Princess Leia, Haley Classic. And this is a really important picture because maybe about 4 days after Bryan got to campus, he somehow was in a long-term relationship with Haley. That's notable because she is his academic sister, and we all know that within lab, intra-lab, romances are strictly forbidden. And so there's that. But then also Haley was apparently in a relationship with some other poor sap who lived somewhere else. So that was and you just gotta love Facebook. So this is Bryan a long time ago. You gotta watch out. Haley clearly does well with the ladies. And then one thing that any of you who know both of them probably noticed is that they love to stick their tongue out. And so I think that it's potentially their own sort of personal code or personal language that they use. And so I noticed that they do like to stick their tongues out too. And then another culture. So maybe they're just really onto something. But all that said, as I said, Bryan has been extraordinarily committed to his project. He's worked really, really hard. He is finishing exactly on date, 2 years from the start to his finish. He's been incredibly easy to work with. He's one of the most naturally analytical people I've ever worked with in terms of a graduate student. I know some of that is Jeremy's grade rubbing off on him, but he's really been a pleasure to work with, and he's now going to talk to you about his master's thesis with you. The title of which is Evaluating Habitat-Based Niche Requirements for Imperiled Bluehead Sucker, Identifying Potential Recruitment Bottlenecks.
Thank you, Phaedra, and thank you everybody for coming to hear about my master's study. Is there a reason the cursor's on me? Well, that's fine. I'll have to deal with that. We live in a world that is anthropogenically altered at a global scale. And whether these anthropogenic alterations change the habitat available for species or they introduce new species, this changes natural ecosystem structure and function. And here we are looking at light pollution at night across the globe, and this can kind of give us an idea of the spread of societal impacts on natural ecosystems and the potential magnitude of those impacts. River ecosystems are particularly threatened by anthropogenic use as they provide and deliver water for societal needs. In this study, the authors determined that river biodiversity is threatened by a suite of stressors that include catchment or watershed disturbance, biotic factors, pollution, and water resource development. And they determined that when these suite of stressors act synergistically, that has the greatest risk to biodiversity in rivers. And they displayed biodiversity threat with warm or red colors indicating higher levels of threat, and cool or blue colors indicating lower levels of threat. And we see a whole lot of red and yellow colors throughout the globe. And they also determined in the study that tributaries are largely understudied, and therefore risks to tributaries are likely underestimated. And tributaries are important to many people in this room as integral parts of study systems of watersheds. One way that these stressors can affect animals at a smaller spatial scale is through recruitment and the life cycle. And for recruitment into the population to take place, the full life cycle must occur. And for a fish species, this might begin with an adult spawning and producing eggs, those eggs hatching into larvae, the larvae rearing into juveniles, and the juveniles eventually rearing into subadults and adults. And for this full life cycle to occur, this requires that complementary habitats exist for each particular life stage to use. Recruitment bottlenecks can occur, however, when either the ability of fecund spawning adults is limited, or the ability of rearing individuals to rear to maturity is limited. And this can lead to suppressed populations, reduced abundances and densities, and lead to concern for the conservation of these populations through time. Some of the potential reasons that recruitment bottlenecks can occur include biotic invasions, food availability, disturbance events, and limited or altered habitat. And that habitat can be physical or thermal alterations. Many of these common threats occur in the Intermountain West, and they include water overallocation with dams and diversions, land use, and non-native species. However, these threats are exacerbated by competition with society for water in this arid climate that we inhabit. Bluehead sucker are an endemic fish in the Intermountain West and have experienced range-wide contraction. They now occupy only approximately 47% of the historical range. In this map, the green points represent where they were sampled and present, and the red points represent where they were sampled and absent. Therefore, these red points represent where they've been extirpated from their native range. And one thing to note from this map is that a large portion of the population exists in the Colorado River Basin. However, there are populations in the Weber River and in the Raft River Basin as part of the Snake River watershed. The population of the Weber River is unique, it's genetically distinct, and these bluehead sucker occupy the Weber River, which is a high gradient alpine river, different from many of the other rivers that it inhabits in the Colorado River Basin. Bluehead suckers spawn on gravel and rear in slow water and backwater areas, and therefore these are some of the complementary habitats that they need for their life cycle to occur. Some populations of bluehead sucker are doing pretty well, such as the population in Farron Creek, noted here with the green circle. This is a tributary to the San Rafael River and later the Colorado River. And Farron Creek has a robust population of bluehead sucker, a relatively depauperate community of fishes, both native and non-native, and it's relatively pristine. It's mostly unaltered flow and thermal regime. And we can use length frequency histograms to evaluate whether successful recruitment is occurring in a population. And here I have frequency on the y-axis and total length or size on the x-axis. And this is the population in Farron Creek. And this population contains many individuals that are in the juvenile, smaller size classes, and fewer individuals when we get out into the larger, older classes. And this indicates successful recruitment into the population, and this indicates a healthy population. This subpopulation from the Weber River, however, is experiencing a likely recruitment bottleneck. With the same axes, frequency and total length, a majority of the individuals in this population are in the adult size class, and this population is missing these juvenile, smaller, rearing individuals. After aging many of these fish, we've determined that many of these adults are 20 years old and older, indicating that successful recruitment into this subpopulation has not occurred for many years, for decades. And if this recruitment population bottleneck were allowed to continue, this would result in the eventual extinction of this population. And in fact, the Utah Division of Wildlife Resources has rescued a subpopulation of bluehead sucker in the Weber River and transplanted them down to a lower elevation reach. The Weber River was more complex historically, and anecdotal evidence suggests that a robust bluehead sucker population inhabited the river with both juveniles and adults. One anecdotal account from the 1920s suggests that over 700 individuals were seined in a single pool. This indicates that suitable spawning and rearing habitats, those complementary habitats, existed to accommodate the life cycle of bluehead sucker. However, large-scale habitat alterations, such as echo and one-chip dams on the main stem, have contributed to alter hydrologic conditions, displayed here with mean daily discharge on the y-axis, across day of year on the x-axis. And here I'm going to compare a reference reach upstream of all major dams and diversions in the upper figure to a downstream reach below or downstream of most major dams and diversions. And when we compare conditions from the 1920s in the blue line to conditions in the 2000s in the red line, we see similar conditions in the upstream reference reach. However, when we compare the 1920s in the blue line to the 2000s in the red line, a significant decline in discharge, especially in peak flow and in base flow across the year in that downstream reach. And these hydrologic changes, in addition to channelization of the river, have contributed to a more simplified geomorphic channel. And currently, the population estimate is less than 500 individuals spread across 84 river kilometers. And when you compare 500 individuals across 84 kilometers to 700 individuals in a single pool, that is a significant and dramatic decline, and poses the question, could this recruitment bottleneck be due to the lack of suitable spawning and rearing habitat? Some alternative hypotheses for why this recruitment bottleneck could occur include competition and predation, for instance with exotic and piscivorous brown trout, and other hypotheses include food and disturbance events. However, my master study is directed at identifying potential recruitment bottlenecks that are habitat-based, and I wanted to evaluate whether habitat availability and quality limit spawning success, and additionally evaluate whether habitat availability and quality limit rearing success. And my study site is the Weber River watershed, located in northern Utah, and the contemporary range of blue-head sucker extends from Echo Dam downstream to the Ogden River Inlet, approximately 84 river kilometers. There was previously a subpopulation in the reach between Echo and Wanchip dams. However, that was the subpopulation transplanted due to a lack of recruitment. Many major dams and diversions occur on the main stem Weber River, and these red stars represent a few of those, including a diversion into the Provo River and some very large dams and irrigation structures that contribute to limit migration of fish and alter hydrologic conditions. And one of the ways that these dams and diversions can have a great impact is through reducing peak annual discharge, displayed here on the y-axis, and it's cutting it off, but across years on the x-axis. And again, I'm comparing this reference upstream reach, upstream of all major dams and diversions, to a downstream reach. And this downstream reach has experienced significant declines in peak annual discharge relative to that upstream reach. And with some of those hydrologic changes in mind, I would like to first talk about my first portion, which is evaluating whether habitat availability and quality limit spawning success. And to do this, I set two sub-objectives: determining habitat characteristics associated with use by spawning blue-head sucker, and additionally evaluating whether spawning and non-spawning habitat differ. I located spawning blue-head sucker primarily by electrofishing large reaches of the river, and we did this by raft electrofishing in the Weber River, and we recorded GPS locations of fish sampled in spawning condition, and then I quantified spawning habitat by measuring habitat characteristics in each spawning reach, and additionally measuring habitat characteristics in 10 randomly selected non-spawning reaches, and then comparing the spawning to non-spawning reaches. Some of the potential issues for studying habitat use in the Weber River include the fact that the blue-head sucker population is a small population. It is already repressed. Their densities are fairly low, so we're not sure that we can expect these fish to occupy critical habitats at saturated densities. Additionally, there is a diverse community of fishes, both native and non-native, that might influence habitat use. And the thermal and hydrologic regime are already altered, indicating that habitat is already altered. And for these reasons, I additionally evaluated spawning habitat use in a surrogate river, and I chose Farron Creek for this comparative approach. Farron Creek, again, has a robust blue-head sucker population, approximately 7,000 individuals in the region that we studied, as well as a proper community of fishes and a relatively unaltered thermal and flow regime. And I wanted to determine whether blue-head sucker would select for higher quality spawning habitat in this river with fewer confounding factors. And when I went out to reaches to quantify spawning habitat, I measured discharge, depths, substrate sizes, channel width, both wetted and bank full, large woody debris at different size classes, and area and depth of geomorphic units. And geomorphic units that we expected to be critical from previous studies conducted on blue-head sucker include riffles, pools, backwaters, and gravel bars. And to analyze these spawning habitat data, I used ANOVA and random forest classification. I'm only going to focus on random forest classification today. I standardized variables by reach length, or as a proportion of the total when that was appropriate, and I categorized substrate based on the Wentworth scale into fines, gravels, cobbles, and boulders. Random forest classification is really useful for highly dimensional data in which the number of predictor variables may be greater than the number of observations. which is really useful for studying small populations such as bluehead sucker, in which sample size is limited. Random forest makes no assumptions about relationships between variables and can handle complex nonlinear relationships. And I use a backward stepwise variable selection procedure, eliminating variables based on variable importance and choosing the final model based on receiver operating characteristic area under the curve. And before I dive into results, I would just like to reiterate that I'm using all of the habitat metrics measured in the field: depths, substrate at different size classes, channel widths, large woody debris at different size classes, and area and abundance of geomorphic units, in order to determine whether what habitat variables are important in predicting spawning or non-spawning reaches, by comparing what we know to be spawning and non-spawning reaches. And in the Weber River, I determined that proportion of cobble, proportion of gravel, and number of riffles are significant predictors of spawning reach classification. And here, the easy way to view the x-axis is by variable importance, with greater values indicating higher variable importance and lower values indicating less variable importance. And the proportion of cobble was the most important variable in this classification, followed by the proportion of gravel, and followed by the number of riffles. And I use partial dependence plots to visualize the trends in these relationships. The proportion of cobble on the top left, proportion of gravel on the top right, and number of riffles on the bottom left. And here the y-axis represents the probability that the model would predict a spawning reach based on average values for all other variables included in the model. And higher values on the y-axis represent spawning reaches, and smaller values on the y-axis represent non-spawning reaches. And proportion of cobble and proportion of gravel at intermediate values more likely to be spawning reaches. And the number of ripples per meter in this case, standardized, at lower values was more likely to be predicted as a spawning reach. This is really important. So in the Weber River, the availability of gravels, cobbles, and riffles are important or significant predictors of spawning habitat. However, I would just like to reiterate the fact that I also wanted to evaluate spawning habitat in Farron Creek, where there are fewer confounding factors. And in Farron Creek now, the proportion of fines, the number of pools, and the wetted channel width were the significant predictors of spawning reach classification, with the proportion of fines as the most important, followed by the number of pools and wetted channel width. And again, I use partial dependence plots to visualize the trends in these relationships. And proportion of fines demonstrated a negative relationship with spawning reach classification, such that reaches that contained fewer fines were more likely to be spawning reaches. And number of pools and mean wetted channel width both demonstrated positive relationships with spawning reach classification, such that reaches with more pools and wider channels were more likely to be spawning reaches. And this is important because in this river with fewer confounding factors, the availability of pools, larger wetted channel widths, and the lack of fine substrate are significant predictors of spawning habitat. And they don't completely concur with results from the Weber River, which I'll get back to later. I would like to move on to evaluating whether habitat availability and quality limit rearing success, the second portion of my study. And to address this goal, I set two sub-objectives: evaluating the relationship between juvenile sucker abundance and size of backwaters in the field, and additionally determining juvenile growth response to different water temperature and velocity treatments. And we conducted the field portion of the study in backwaters through the contemporary range of bluehead sucker, and we sampled backwaters within and immediately downstream of known spawning reaches. And these backwaters varied greatly in size, from approximately 10 square meters to 300 square meters. And I located juvenile suckers by sampling these backwaters and counting all sucker species synonymously. Similar to some of the concerns in the spawning habitat portion of the study, we expect that the reduced densities of rearing bluehead sucker juveniles would not allow us to evaluate their suitable rearing habitat with bluehead sucker alone. And we also expect that the other sucker species in the Weber River, which include Utah sucker and mountain sucker, two native species, would be ecologically synonymous with bluehead sucker at this small size. And I measured habitat characteristics such as area and depth in these backwaters. And I conducted this portion of the study over 2 years by seining 18 backwaters in 2015 and backpack electrofishing before seining 11 backwaters in 2016. And we conducted this more thorough sampling in 2016 in an attempt to reduce the size bias of seining only, and also to elucidate the effect that larger fish might have on rearing habitat use of sucker juveniles. And 10 of the backwaters were sampled in both years, 2015 and 2016. To analyze these data, I used a linear mixed effects regression and a forward bidirectional variable selection procedure. And I wanted to evaluate whether the predictive variables of maximum depth, area of backwaters, and brown trout abundance could significantly predict the sucker sampled in a backwater. And I took into account the random effects of year and site because we sampled over multiple years and we resampled some sites. And we would expect that size would be related to rearing sucker habitat use. And we found that maximum depth was our one significant predictor in this model, with maximum depth displayed on the x-axis and total sucker sample displayed on the y-axis. And the raw data suggest a positive relationship, and the model confirms that positive relationship. Backwaters that are deeper are significantly related to or provide rearing habitat and are used by more rearing suckers relative to shallower backwaters. And this is important because a positive relationship exists between depth of backwaters and rearing sucker abundance, and these backwaters may be largely depleted through the Weber River main stem based on hydrologic conditions. To evaluate my second subjective, determining juvenile growth response to different water temperature and velocity treatments, I would first like to point out a concern that we had moving into the study, and this is the altered thermal regime and thermal effects on bluehead sucker. Few studies have been conducted on bluehead sucker, but one suggests that the optimal temperature for juvenile growth is 17.5 degrees, displayed here with this blue line, and with mean daily temperature on the y-axis and time across the growing season as months on the x-axis. The black line represents the temperature of a middle elevation reach through the summer of 2015. And we can see that the temperature in that reach did not come close to that optimal temperature of 17.5 degrees until approximately September, when it was still below that level. And we expect that this is largely due to hypokinetic dam releases, reducing temperatures in the Weber River main stem. And to address some of these concerns, I conducted laboratory experiments to evaluate juvenile growth at varying water velocity and temperature treatments. And we conducted these experiments in steel stream flow-through tanks. I collected juvenile individuals, juvenile bloodsucker, from the Raft River, which is in the part of the same ESU or evolutionarily significant unit as bloodsucker from the Weber River, indicating that they are more genetically similar to Weber River bluehead sucker than they are to bluehead sucker from the Colorado River Basin. And I collected 140 individuals. I set temperature and velocity treatments based on the range of conditions encountered in the Weber River and also from the literature, which is largely conducted in the Colorado River Basin. I set 3 velocity treatments, slow, medium, and fast, and 3 temperature treatments, cool, tepid, and warm, for a total of 9 treatments. I fed all fish to excess, I did not reuse any fish, and I weighed and measured each fish before and after treatments in order to determine growth. And in these experiments, I was testing 2 hypotheses. The first of which is that slower water is optimal for juvenile growth. We know that in faster water, energetic cost to fish can be greater, meaning they may grow less. And we also tested the hypothesis that warmer water is optimal for juvenile growth. We know that in warmer water, metabolic processes in fish can increase, meaning that if food is not limited, fish may grow more. And to analyze these data, I used a linear mixed effects regression, evaluating whether the predictive variables of water velocity, water temperature, and the interaction between velocity and temperature could significantly predict growth measured as grams per day. And grams per day is grams of growth through the treatment divided by the mean weight of the fish through the trial divided by the number of days. And I took into account the random effects of tank nested within trial. And I would first like to display results of growth as grams per day on the y-axis across velocity treatment on the x-axis measured as tank discharge, with the colors representing temperature, with blue being the cool temperature treatment, green the tepid temperature treatment, and red representing the warm temperature treatment. And when I plot model predictions for each temperature treatment, we see that growth declines across velocities for the cool temperature treatment and decreases across velocities for the tepid temperature treatment, and remains pretty consistent across velocities for the warm temperature treatment. And when I include standard error to those predictions, we see that they largely overlap, and fish growth is equivalent across velocities, again for the warm temperature treatment, and not significantly different across the tepid temperature treatment. However, in the cool temperature treatment, in this blue polygon, growth is significantly higher at slower velocities relative to faster velocities. And this is important because these juvenile bluehead sucker are experiencing increased growth in slower water at cooler temperatures, especially. And next, I would like to display results of growth in grams per day across temperature treatments. And the different colors and line types will represent the velocity treatments of slow, medium, and fast. And when I plot the model predictions based across velocity treatments, we see a decline in growth across temperatures for the slow velocity treatment, and a decline in growth across temperatures for the medium velocity treatment, and a slight increase across temperatures for that fast velocity treatment. And when we include standard error, juvenile bloodsucker growth is equivalent across all velocity treatments in the warmer temperatures. However, juvenile bloodsucker growth is significantly greater in the cooler temperatures at slower velocity relative to faster velocity, where there is no overlap between the two polygons. And this is important, and this suggests that the optimal temperature may be cooler than we hypothesized for juvenile bluehead sucker growth. Based on that previous study, we expected the optimal temperature for juvenile bloodsucker growth to be approximately 17.5 degrees, with temperature on the x-axis and growth from low to high on the y-axis. However, we determined that the optimal temperature appears to be much cooler. It appears to be much closer to 12 or 12.5 degrees than 17.5 degrees. And one important point to note is that the optimal temperature can be higher with greater food availability in the wild, although we did feed these fish ad libitum in the experiments. And one really neat thing about using a linear regression is that we can use the model parameters and coefficients to predict growth across time. And I use this to predict the weight of a fish or growth of a fish across an entire growing season of 120 days on the x-axis for a fish that existed or inhabits a cool temperature, slow velocity stream with the blue line, a tepid temperature, medium velocity stream with the green line, and a warm temperature, fast velocity stream with the red line. And visualizing the growth across an entire growing season, we see that the fish in the cool temperature, slow velocity stream grows significantly more than either the fish in the tepid temperature, medium velocity stream, or the warm temperature, fast velocity stream. And this may have important implications for the survival of these fish and the eventual recruitment into the population. To summarize, bluehead sucker are endemic to the Intermountain West. They've experienced range-wide decline and are imperiled range-wide. The population of the Weber River is genetically distinct and at risk of extinction due to a lack of recruitment. I conducted this study to determine habitat associated with use by spawning bluehead sucker. And somewhat unsurprisingly, the results did not completely concur between the Weber River, in which population densities are low and habitat is already degraded, and Farron Creek, in which population densities are high and habitat is fairly pristine. And in the Weber River, gravels, cobbles, and riffles, rather, were significant predictors of spawning reach classification. And gravels and cobbles are concurrent with what we'd expect based on the fact that bluehead sucker spawn on gravel. However, the negative relationship between riffles and spawning reach classification is contrary to what we would have expected. In Farron Creek, however, where there were fewer confounding factors, the lack of fine substrate and the availability of pools and wider channel were significant predictors of spawning reach classification. And these are consistent with what we'd expect based on previous studies of bluehead sucker. And the differences between the two rivers and the fact that the results did not completely concur suggests that the realized niche of bluehead sucker in the Weber River may be confined by biotic factors. And a potential source of that is non-native brown trout, piscivorous brown trout. And we sampled this brown trout consuming another brown trout. And the predator brown trout was a common size for adult brown trout in the Weber River. And the prey item was approximately 300 millimeters total length, which is just smaller than an adult bluehead sucker, which poses the possibility that fish such as brown trout could be influencing habitat use in the Weber River. In the field, I determined that deeper backwaters are associated with use by more Riverian sucker. However, the contemporary hydrologic regime and channelization limits the slow-water Riverian habitat in the Weber River main stem. Here, some of the channelizations displayed are riprap on the bank and cars dumped into the bank. And so the reduction in peak annual discharge and channelizations likely limits the currents of backwaters through the Weber River main stem, and certainly limits the occurrence of large deep backwaters through the Weber River main stem. In the laboratory, I determined that slow velocity, cooler temperature treatments are more optimal for juvenile growth. And this is consistent with what we'd expect for slower velocity relative to growth. However, this is a contradiction to previous research for the optimal temperature and suggests that there might be a local adaptation to temperature in the more alpine Weber and Rapp rivers relative to where most studies have been conducted in the Colorado River Basin. This also suggests that lower temperatures from dam releases may not be as much of an issue as we previously expected. However, diversions in dewatered reaches, or reaches below diversions and dewatered reaches, are likely much warmer than they would be naturally. And elevated flows from dams maintain high flows through the irrigation season to reach downstream irrigation customers and certainly reduce slow water or slow velocity habitat in the Weber River. So I've identified some potential recruitment bottlenecks that are habitat based. And we know that contemporary hydrologic conditions may limit the availability of these critical spawning and rearing habitats identified in the study. But I hope that targeted rehabilitation may allow for recovery, and this could be conducted at either the watershed scale or reach scale. At the watershed scale, this might include restoring natural hydrologic conditions or restoring an approximation of natural hydrologic conditions, which would facilitate the creation and maintenance of complex in-stream habitat, and also promote riparian recruitment and thus large woody debris recruitment into the system, creating a cycle of self-sustaining in-stream habitat. One important point to note is that this does not need to occur every year, especially for a long-lived fish such as bluehead sucker. At the reach scale, restoration might include avoiding dewatering critical reaches, especially after successful recruitment. This might include adding gravels and cobbles, particularly downstream of dams where these substrate sizes may be limited, and the manual creation of slow water habitat. And this is an image of a restoration plan directed at creating slow water habitat in the lower Weber River. And one really exciting part of this project is that restoration efforts have been directed at bluehead sucker and directed at improving habitat for bluehead sucker in previous cases, such as in the San Rafael River drainage downstream of Farron Creek. And I'm fortunate to work with Bryan in the lab, who helped create this science-based restoration plan. And I think it would help restoration plans directed at bluehead sucker in the Weber River to follow some of the plans developed for this species elsewhere. In the San Rafael River, some restoration actions proposed included removing non-native vegetation, and also facilitating dam building activity by beaver. And I'm not necessarily saying that these are the solutions in the Weber River. However, the potential benefit and the goal of these actions was to enhance natural river processes, and I think that restoration of the Weber River would benefit from attempting to do that. And possibly the most exciting part of this project is the existence of the Weber River Partnership that has largely funded this master study. And the Weber River Partnership includes many different stakeholders throughout the basin, such as irrigation entities, water conservation districts, Trout Unlimited, Pacific Corps. So a bunch of different players coming together and funding the study. And we are dealing with a degraded river, a sucker species that is generally perceived as non-charismatic, of no or low economic value and no recreational value. And it's great that the Weber River Partnership has come together to fund this study and will likely fund restoration actions directed at this population and directed at recovering the population. And with that, I'd like to thank some of these many funders, including the Division of Wildlife Resources, the City of Ogden, Davis and Weber Counties Canal, Pacific Corps, Provo River Water Users Association, Trout Unlimited, Weber Basin Water Conservancy District, Weber River Water Users Association, Bureau of Reclamation, the USGS Co-op Unit, and the Ecology Center. And many in that list, Davis and Weber Counties Canal, Pacific Corps, Provo River Water Users Association, Trout Unlimited, Weber Basin Water Conservancy District. I mean, these are all entities that you would not expect to direct money towards a sucker species and their habitat. I would like to give an especially warm thank you to the DWR Northern Region Office, including Paul Thompson and Chance Berdarius, who are here today. Paul has been instrumental in this study and getting this rolling. I'd also like to thank the field technicians that have helped me through this project, which include Thomas and Caitlin that are here today, as well as Brandon. And I'd like to thank Joe and Wally from the FHC, and everybody in the fish ecology and lake ecology lab. And I would like to thank Phaedra, who has been a great advisor through this process and has been really patient with my development through this study. Phaedra emailed back from Mexico after seeing my thesis proposal and wrote, "Bryan, this was a great start. You have some work to do, however. It reads a bit like a Harlequin romance. You need to learn," and I don't know what it said next, but possibly everything. and Phaedra has really helped me through that process in the study, guiding my energy to the right sources. I would also like to thank Jeremy, who has been instrumental in helping me think about ecological questions and analyze data critically, and has certainly gotten me to root for Wisconsin more than I would like to admit. And I would like to thank Gary, who has helped me greatly with logistics and field operations and taught me a million things, such as how to write multi-tiered emails that last pages, and also to recognize the fact that while waders are waterproof, they are not often soundproof. And I would like to thank all of my friends and family that have made this process so much fun. And with that, I can take any questions.
Given how expensive and uncertain engineering solutions sometimes are, is there any interest from the water district or any of your collaborators on, for instance, removing brown trout from a 1 mile section of river as an experiment?
Not that I have heard of. One thing I should have mentioned is that there's a follow-up study to mine which will evaluate the potential impacts of brown trout on bluehead sucker through diet and isotope data, which will likely come after a hatchery broodstock is available for bluehead sucker, and there are actually bluehead sucker in the river that brown trout could possibly be eating, whereas now the densities are so low that it's unlikely to detect any influence they might have. But that's a really interesting suggestion. It's certainly one that I've thought of. Considering the fact that there are so many barriers for migration, it wouldn't be that hard. Well, it would be fairly difficult, but it's feasible to have one reach of brown trout-free river. I'm not sure how interested all the parties are in that, considering that the brown trout fishery is prolific and worth money.
So you saw that portion of fines made a difference on Farron Creek. So on the Weaver River, were there fines there? Was there variation in fines? Is it possible that it would be important in the Weaver if they were present?
So the lack of fines were important in Farron Creek for spawning habitat.
Right.
It was a negative relationship. And I think that is somewhat inherent in the fact that the gravels and cobbles were significant predictors in the Weaver River, considering that those aren't fines. So I think that some of that is represented in that relationship.
I was wondering if it's a difference in rock type or something, and whether Farron Creek had more fines in it, such that there were places that it would suppress the fish and others. That's why I'm asking whether there were fines in the Weaver River.
Well,
There were
usually different systems, right? There's a lot of sands in Barron Creek, right?
Yeah, yeah. I mean, there are plenty of sands in Farron Creek. There are also a lot of fines in the Weaver River, though. And depending on the reach that you're in, I mean, it's a fairly different river at different sections based on dams and diversions.
I was curious if you had a site on the Weaver River that was similar to the Farron Creek site in terms of channel geometry, because it seems like, you know, if the Farron Creek is a smaller river, that could tell you why width was important, and that might not transfer directly to the Weaver in sort of dimensions and not hydrology, the whole thing is different.
Yeah, and we selected Farron Creek partly because it has a robust bluehead sucker population, but also partly because it is fairly similar to the Weaver River, especially in the canyon confined reaches of the Weaver River. And I expect that I guess I expect them to be fairly similar in that aspect. And an additional analysis included, that I didn't display, included comparing the rivers, you know, spawning habitat in Weaver River to spawning habitat in Farron Creek. But yes, I'm not really sure. I mean, they're fairly similar gradient, especially in certain reaches. Is that really?
So I'm sorry if I missed this, but outside of biodiversity that this fish contributes to these rivers, what are some ecological functions that they might serve? These reductions in numbers that we found, have there been some reduction in the ecosystem services that this fish provides?
Yeah, so that's a really interesting question, and that's something that I thought about, especially since Michelle Marbier was here this week talking about biodiversity. Unfortunately, it's a fairly understudied fish. It's not of economic value based on angler interest. So I think for that reason, there have been few studies conducted on the fish. So I don't think we necessarily know to what extent it provides ecosystem services. And I think currently with reduced densities, I'm not sure that we could really evaluate that at all, especially in the Weaver River. But I mean, it's a sucker, so it, you know, it has cartilaginous scraping ridges on its mouth. It consumes some invertebrates and algae, so it may be instrumental in nutrient cycling.
I was just curious about, so you said as far as restoration, so in the Weaver, increasing backwater habitat would be key. And so you brought up the fact that maybe increasing flows would do that. Can you expand on that a little bit? Because that seems foggy to me, is like how that would happen.
The idea would be to increase peak discharge in order to create some slow water rearing habitats and some in-stream complexity. I'm not sure that's how viable that solution is in the Weaver River, but that would be a solution in which the river would create its own habitat rather than going in with backhoes and hard engineering structures. And one of the issues with that is, the time frame it would take to acquire the water rights and everything in order to actually, have environmental flows would be a pretty long time. And so, hard engineered structures, might possibly be the best solution in this case, because this is a fish that, could go extinct in a decade or a couple years, or, it's at a smaller time scale.
So it's not uncommon for really old fishes to, while they're sexually mature, they don't exhibit spawning behavior and they just reabsorb, especially females reabsorb the eggs. So how do you know, given how old this population is, given that they're close, the individuals are close to senescing out, how do you actually know that a spawner equals spawner reach, and that the characteristics that you're finding are actually where they are spawning habitat?
I guess the short answer is that we cannot be 100% certain about that, but we expect expect to be more confident as sample size increases. And to answer the kind of first part of your question, the fact that many of these fish are older and might not be reproducing every year, many of the fish that we studied, or many of the reaches that we surveyed for spawning, bloodsucker and spawning condition, were much younger than the subpopulation that I displayed on the length frequency histogram. So many of the individuals were actually 3 to 5 years old from the spawning surveys last year, and from the previous year, 5 to 7 years old. So a lot younger based on what we've aged in the lab.
Well, there's no more questions. I do want to tell people that we are going to converge at the White Owl at 6 p.m., assuming that the next section goes well. And we'd love to see any of you there. And I will be buying beers for a while. Yeah.